Hello students, this is Dr. Ranjini, Assistant Professor in Biotechnology. Today we learn about structure and functions of ribosomes. Ribosomes are protein synthesis units of a cell. These were described by G. E. Pellade in 1952. They are the complex of ribosomal RNA and various proteins. They are present both freely in the cytoplasm and they are adhered to the surface of endoplasmic reticulum. And the presence of these ribosomes on the surface of the endoplasmic reticulum has been proved by electron microscopy. Ribosomes are small, dense, rounded and granular particles of ribonucleoproteins. They occur in most prokaryotic and eukaryotic cells and provide a scaffold for the ordered interaction of all the molecules involved in protein synthesis. The small proteins of 100 to 200 amino acids, therefore, made in a minute or less. On the other hand, it takes 2 to 3 hours to make largest known protein, titin, found in the muscles and contains about 30,000 amino acid residue. So it takes minute or less to prepare the very small peptides of around 100 to 200 amino acid, but it takes 2 to 3 hours to make largest known protein, or the largest protein synthesis require at least 2 to 3 hours. Coming to the occurrence and distribution of these ribosomes, they occur in both prokaryotic and eukaryotic cells. In the prokaryotic cell, the ribosomes often occur freely in the cytoplasm or sometimes as polyribosomes. In the structure of the E. coli, we will find the chain of ribosomes. So these ribosomes will be attached to the string of RNA and they are called as polyribosomes. So such type of polyribosomes will be observed in prokaryotic cells. In eukaryotic cells, the ribosomes either occur freely in the cytoplasm or remain attached to the outer surface of membrane of endoplasmic reticulum. In eukaryotes, they are freely present in cytoplasm or they are bound to the surface of endoplasmic reticulum. Coming to the types of ribosomes, ribosomes are classified into two types based on their sedimentation coefficient, that is 70S type and 80S type. Here S stands for Swedberg unit and it is related to the sedimentation rate. That sedimentation, the rate at which it will settle down or sediment, depends on the mass and size of a molecule. Thus the value of S indicates the size of the ribosome. 70S ribosome. Now we will learn about 70S ribosomes. Prokaryotic cells have 70S ribosomes. These 70S ribosomes are comparatively smaller in size and have a sedimentation coefficient 70S with molecular weight of 2.7 into 10 to the power of 6 Daltons. The electron microscopy measures the dimension of the 70S ribosomes as 170 into 170 into 200 angstrom unit. They occur in prokaryotic cells of blue-green algae and bacteria and also in the organelles like mitochondria and chloroplasts of eukaryotic cells. Coming to the 80S ribosomes, eukaryotes have 80S ribosomes. The 80S ribosomes have sedimentation coefficient of 80S with their molecular weight 40 into 10 to the power of 6 Daltons. The 80S ribosomes occur in eukaryotic cells of plants and animals. The ribosomes of mitochondria and chloroplasts are always smaller than 80S cytoplasmic ribosome because they have 70S ribosome. and are comparable to prokaryotic ribosomes in both size and sensitivity to antibiotics. So how many number of ribosomes will be there in a cell? So here an E. coli cell contains around 10,000 ribosomes, forming 25% of total mass of the bacterial cell, whereas mammalian cultured cell contains 10 million ribosomes per cell. You can imagine the complexity here regarding the number. So more numbers are present in the eukaryotic cell compared to the prokaryotic cell. Here in E. coli we will find 10,000 ribosome, but in case of the mammalian cell we have 10 million ribosomes. So what about the chemical composition? So this ribosome is having 60 to 65% of RNA, and when you see the different subunits like 50S subunit, it is made up of 23S rRNA, 5S rRNA. So we all know the ribosomes are the combination of RNA and the protein. So they are the ribonucleoprotein. So 50S subunit will be having 23S rRNA, 5S rRNA. Whereas 30S subunit of the 70S subunit type of the ribosome will find 16S rRNA. So that is regarding the 70S ribosome composition. Coming to the 80S type of the ribosome, it has the larger subunit, that is 60S, and the smaller subunit 40S. And if you see the chemical composition of 60S subunit, it is made up of 28S rRNA, 5S rRNA and 5.8S rRNA. The composition of the larger subunits of each 70S and 80S type is different. And then 40S subunit of 80S type of ribosome is having 18S rRNA. So that is regarding the different type of RNA we will find in the 80S type and the 70S type of ribosome. In each ribosomal subunit, RNA is in the form of highly folded filament. And to this many proteins are adhered, many proteins are attached. And 60% rRNA is in double helix form. Though it is folded filament, it is a double helix form. Most abundant nitrogen bases are guanine and cytosine. You know the difference between RNA and the DNA. The DNA is having AGCT nitrogen base, whereas in case of RNA we will find the AGCU. Among that, the rRNA here, it is rich in guanine and cytosine. The ribosome also contains the metallic ions. The most important low molecular weight components of ribosomes are the divalent metallic ions, and this ribosome has the more of Mg2+, Ca2+, and Mn2+ divalent cations. They play an important role in the integrity of this larger subunit and the smaller subunit. The aggregation of these subunits will be due to divalent cations. This is regarding the structure of the ribosome. As I was telling earlier, whether it is the 70S type or the 80S type of the ribosome, ribosomes are oblate spheroid structures of around 150 to 250 angstrom in diameter, and each ribosome is porous, hydrated, composed of two subunits. It is composed of two subunits. One ribosomal subunit is larger in size. It is larger in size, and it may be 60S subunit in case of 80S ribosome, or it may be 50S subunit in case of the 70S ribosome. So the subunit which is larger in size has a dome-like shape. It has a dome-like shape, while the subunit which is smaller in size, occurring above the larger subunit, or it is present adhere to the larger subunit. The smaller ribosomal subunit contains the single rRNA molecule, referred to as small rRNA. The large subunit contains a molecule of large rRNAs and one molecule of 5s RNA plus additional molecule of 5.8s RNA in vertebrates. We saw in the composition of 50s, 80s and the 70s type of the ribosomes. And there are great structural and functional similarities between 70s and 80s type of the ribosomes. Though they differ in their size, they show the similarity in their the structure and the function. And these 70s and the larger subunit and the smaller subunits are held together by the divalent cations. So if the level of these Mg2 plus ions comes down, the separation of the larger and smaller subunits takes place. Actually, in bacterial cells, the two subunits are found freely in the cytoplasm, and they unite only during the process of protein synthesis. At high concentrations of Mg2 plus ions in the matrix, usually the two ribosomes will be associated. But in case of the bacterial cell, we'll find them freely floating in the cytoplasm, but during the synthesis of the protein, they unite. But the divalent cations, or the Mg2 plus ion, play an important role in the aggregation or the binding the larger subunit with the smaller subunit. During protein synthesis, many ribosomes are aggregated due to common messenger RNA and form the polyribosomes or polysomes. So to the chain of the so whatever we are seeing here like this, we'll find the mRNA to which the ribosomes or the larger and the smaller subunits will be attached, and many the ribosomes will be attached on its surface, and this structure is called as the polyribosomes or polysomes. The actual three-dimensional structures of bacterial rRNAs from thermopolis recently has been determined by X-ray crystallography. So they have studied the X-ray crystallography structure of 70s ribosome. The much more ribosomal proteins for the most part are associated with the surface of the rRNA. During translation, the ribosomes moves along an mRNA chain, interacting with various protein factors and tRNA, and are very likely undergoing large conformational changes. So during protein synthesis, they will undergo various the changes in their conformation, their appearance. Despite the complexity of the ribosome, the great progress has been made in determining the overall structure of the bacterial ribosomes and in identifying various reactive sites. And this ribosome, which is involved in protein synthesis, will be aggregated around the mRNA, and the tRNA will help in synthesizing the protein. So that is regarding the structure of the ribosome. Now we'll move on to the functions of ribosomes. Ribosomes assembles amino acid to form the protein. So one after the other, the amino acids are joined together by peptide bond to form the protein. And all the amino acid, they look like the chain of beads. And they assemble the amino acid to form the functional protein, and they are essential to carry out various cellular functions. The DNA produces mRNA by the process of DNA transcription. We know about central dogma of the cell, where DNA will be converted into RNA. Then from the RNA there is formation of the proteins. There is formation of protein. So any function in a cell is mediated by protein synthesis. But the signal or the message for this protein is from the DNA. So this is called central dogma of the cell, wherein DNA gets converted into RNA by the transcription, and then RNA will encode the protein to carry out the particular function inside the cell. The mRNA synthesized in the nucleus is transported to the cytoplasm for various protein synthesis to carry out various functions. The ribosomal subunits in the cytoplasm are bound around mRNA polymers, and they form the polysomes or the polyribosomes. The tRNA then synthesizes the proteins. The proteins synthesized in the cytoplasm are utilized in the cytoplasm itself, and the proteins synthesized by the bound ribosomes are transported outside the cell. So cytoplasm, freely floating ribosomes in the cytoplasm will synthesize the protein and it will be utilized in the inside the cell itself. But the bound ribosomes on the endoplasmic reticulum, rough endoplasmic reticulum, will be transported outside the cell. This is regarding the structure and functions of ribosome. I hope you understood today's virtual class. Thank you.